In this section of the lecture, we're going to talk about two different types of bone growth. In the beginning, we're going to focus on two types of bone growth that occur during the embryonic and fetal development. Later, we're going to talk about type of bone growth that occurred during the infancy up to the adulthood. Okay, so the two types that we're going to talk in the beginning, they are called intramembranous ossification or inside membrane, and endochondral ossification, or is the one that occur inside of the cartilage. Okay, so these two types of ossification are very interesting because although both form, the end result is the formation of bones. One start using loose connective tissue, while the other one start using cartilage. However, both converge into the production, into the formation of bone. Then we will move into the two different types of bone growth that occur during the lifetime. So when you grow, yeah, when you get older, when you have to repair your bones, and we have two types. One type that is the interstitial bone growth, that is the one that occurs when you grow in length, and the other one is the appositional bone growth, that is the one that occurs when you grow in width. Yeah, so here, very clear, bone growth in length, and this one is that when your bones has to grow in diameter, yeah, so in width. So let's start. So here we're going to start talking about bone formation, and you will hear two similar topics, osteogenesis or ossification. Both in this class we're going to use it synonymously as the process of bone formation. And remember, if you remember a few classes ago, if you go back to your notes, we told you that the connective tissue form from these mesenchymal cells. So these stem cells will start forming, will form, differentiating to completely different tissues: cartilage, bone, tendons, irregular connective tissue. And the difference is that they differentiate using different transcription factors, yeah, and also cues from the environment. So in the case of the chondroblast and osteoblast and the formation of cartilaginous bones, although they share very similar pathways, the cues like the amount of oxygen will be fundamental to form cartilage or form a bone. Okay, so remember, during this lecture I will talk about the process of bone formation, and bone formation originate from a connective tissue. So all of them will form from a mesenchymal stem cell. And then there will be a process of differentiation that require the expression of particular genes. And there will be two key terms that here for this class we are going to use synonymously: osteogenesis and ossification. And both mean process of formation of bone. Now we're going to start talking about bone formation, and I have this diagram of your textbook that I found that is very useful. Hopefully at the end of this class you will be able to recognize that in the fetal stage there are different type of ossification. So here, if you remember, here there's one intramembranous ossification, there is also endochondral calcification, and here you can see that in the skeleton in the beginning you have cartilage. Okay, so if you see, there are different colors there. You see white, you see clear light pink, and you see darker pink. All of them are different type of structures. Yeah, some of them will be bones, some of them will be connective tissue, some of them will be cartilage. And hopefully at the end you will be able to detect which one is which and when they start appearing. So first thing that I have to ask you is, when do you think that the bone should start? It should start right away, or they should happen gradually? And should you protect some structures better than other ones in the beginning? So for example, do the brain need to be protected right away? So um, during our skeleton start forming just first week after conception. So there are parts of the skeleton that are already forming. Okay. However, they don't form right away into bones, most of them. Yeah, so there are bones that start forming, but there are other ones that only form cartilage and other ones that only form some type of connective tissue. Yeah, later on, and in this case, after 8 weeks after conception, you start ossifying, start the process of ossification of some cartilage and some connective tissue. Okay, so this is before birth, and you have dramatic changes here. However, during adulthood, you still have to continue creating bone. Yeah, not as dramatic as before, but depending on your lifestyle, you will have different type of bone formation. So while I delete all these things, I wonder you think, what happened if you go to the moon? Have you heard what happened when with the bones with somebody that goes to the moon and there is no gravity? Yeah. So what happened is that when we go on, if there are astronauts that go to space, spend 6 months there, and they come back, they have lost bones there. Yeah, they have lost some amount of bones because as they are not, there is no gravity. The bone formation start changing, so they feel that they don't need as much bones. And the same thing happen more gradually if we have a sedentary life. Your bones start also changing. Okay, so here I'm showing you what happened. Maybe some of you play a sport. Hopefully, none of you these things had happened to you. But however, if you play sport, I'm sure that one of you had a fracture, and there are different type of fractures. Yeah, so there are some fractures that break the skin, so you can see blood. And other ones, there you will see some structure that some fracture that the bone is broken, but there you don't see blood. However, every time that you have a fracture, the bone breaks. And if you ask me, bones have blood vessels and also and the nervous system and neurons around it. And a person who has ever break a bone will say, yes, definitely, it hurts a lot. Yeah, they have different. The nervous system is there. Yeah, so what they have to do now is to repair. Yeah, so they have to create new blood vessels. They have to create the nervous system have to be completely repaired. So you need stem cells even in the adult stage to completely heal the bones. Okay, and this can take from a few weeks to a few months. Okay, so I told you that the bone formation, the bones are formed in 4 different situations. In the beginning, I told you that during embryological and fetal development is very important that you start, that not all the skeletons start forming at the same time, that it start in a gradual process, and in some cases you form cartilage and you form connective tissue that later on is going to be ossified, is going to be calcifying and forming to bone. I also told you that after birth, um, you start also changing. So, for example, uh, when you go from being a kid to be an adult, you have to grow. So, this is another way in which the bone have to be formed. In addition to that, and I focus on the bones remodel, I told you that the bones also have to be remodeled, and I give you an example of the astronauts, yeah, and also about having a sedentary life. In that case, also the bones have to be reformed. And finally, if you have an injury like a fracture, the bones have to be healed. So you need stem cells, and you need to repair and form new bones. Okay, so 4 different from different stages or situation when bones are formed. This is occur after birth, and this one occur before birth. Okay, and here we have a key cell for the bone formation, and we talked before. So you remember the osteoprogenitor cells. So they check where they are located. They can be located in different places, and in the periosteum and the osteum, close to this Boltzmann canal, all close to blood vessels, you will find them there. And their function, if a bone break, they're the stem cells, so of the bone, so they're going to differentiate into osteoblasts that are the ones that form the matrix. They're going to secrete a lot of and start the process of calcification, and then they form osteocytes that they maintain the tissue. So remember, the osteoprogenitor cells play a key role during bone repair and growth. Yeah, um, they are the key in this process because they allow the formation of all these other cells. Just as a reminder, although these one all have a similar origin, there is another type of cell in the bone tissue that has an immune origin. Yeah, so that are the osteoclasts that are also important during the bone formation, and more than the bone formation in the bone repair. Yeah, they destroy the bones that are not working very well and allow that a healthy bone appear. So just to recap, I remind you that the osteoprogenitor cells are the stem cells of the bone. They are also called osteogenic cells. They are in charge of bone repair and bone growth. And they are the precursors for specialized bone cells like the osteoblasts and the osteocytes. And here are ready to talk about these two guys. So to finish this section of the introduction, I wanted to tell you another function of the osteoprogenitor cells. So if you remember, they are located in this perichondrium or periosteum. And I told you that there are two types of cells. The one that have the suffix blast, that are the young cells, and the cytes that are more mature cells. Yeah. So you have two types. I told you about osteoblasts, but you can also have chondroblasts. And I told you about osteocytes, but you can also have chondrocytes. Yes. One are cartilage, the other ones are bones. So what is the difference between these two structures? How do you start turning bones into, cartilage into bone? Yeah, so it's pretty easy. There is a chemical cue that can tell the cells turn into a cartilage or turn into a bone. And the main cue is the amount of blood supply. If there is a low blood supply, they will form, the osteoprogenitor will form chondroblast and chondrocyte. However, if the blood supply is high, they will form not chondroblasts, they will form osteoblasts, and they will form osteocytes. Now we're going to move into the first type of bone growth that occur during the embryonic stage, the intramembranous ossification. Remember, in this one, we will have the loose connective tissue calcifying and forming a bone. Okay, so let's start with that one. So remember, two types of ossification, and here we have the two types, intramembranous and endochondral. So there is first a process of divergence. Both come from the mesenchymal tissue, yeah, from mesenchymal stem cells. So in that case, both have the same precursor. However, the path that they take is very different. One becomes loose connective tissue and the other one becomes cartilage. However, both converge into the same tissue, that is the bone tissue. So they have a similar precursor, but they have different ways, in different pathway, different genes to converge into a bone tissue. Another difference is how fast they ossify and which bones they ossify. So, for example, the first one that you see here in purple, the intramembranous, ossified the skull, the clavicle, on most of the cranial bones. So here you can see in this schematic the skull, clavicle, on all these cranial bones that are very important because they need to ossify fast because the brain need to be protected. So remember the intramembranous ossification, like in the case of the skull, they need to ossify fast. On the other hand, here you can see that most of the skeleton come from an endochondral, from a cartilage origin, and these are axial skeleton and long bones. So all the ones that here that are in blue, the long bones, the humerus, the pelvis, the femur, all of those ones have an endochondral ossification, and those one ossify later. Okay, but very important, as I said, they're keep better bounce, not break. So the idea is that in the embryonic origin, there is some flexibility, but later when they grow, you start seeing that they need to ossify. So now let's start with the mechanism. What are the processes that happen for the formation of bone using this intramembranous ossification? And here you have the step one in your textbook. They remind you that flat bone of the skull and the mandible follow this type of ossification. And here they show you this schematic to simplify everything. First thing that they tell you is that the first step is the development of a ossification center. And here in white, all this surrounded this area is the ossification center. And in the ossification center, close, there will be, of course, blood capillaries. In gray, you will have, or light blue, you have all these collagen fibers because there is a loose connective tissue there. And in the ossification center, you start having the first osteoblast. So here you see one osteoblast here, and they start secreting the organic matrix to start forming this ossification center. And in addition to that, you have all these mesenchymal cells. Then they need to migrate. Yeah, they will migrate and go to the ossification center to start forming not only osteoprogenitors but osteoblasts and start secreting more and more extracellular matrix. So in the second step, after the formation of the ossification center, that you can still see here, so the ossification center is there. However, there was a dramatic change. And if you see now, you see this brown area. And if you see the osteoblasts, now they are located in the outer side of the ossification center. And inside, so look here, you can see the osteoblast. Yeah, and you can see that inside you have a newly calcified bone extracellular matrix. And inside you don't have any more osteoblasts, what you have is osteocytes. And you can see them forming the space that where they're going to be. That is going to be the lacuna. And in addition to the lacuna, you can see the formation of the first canaliculi that start forming there. So remember, in the step 2, we took the ossification center and we started the calcification in that ossification center. And what we did is, or what what happened there is that there was calcium and other minerals that are being deposited in that area. And all these extracellular matrix start becoming harder, yeah, because it is calcified. So we already form an ossification center, then we calcify it, and finally we form the spongy bone trabeculae. Okay, so let's see the diagram, and we see that still the connective tissue is there, the loose connective tissue with all the fibers, but just from the mesenchymal cells, you see that they are condensing, so they are ready to differentiate into another cell. Then you see that the blood vessels, they are not only in the connective tissue, but they start going in the spaces between the bone. Yeah, so here you see that the extracellular matrix develop into a trabeculae, yeah, into these spiny pieces of bones. Yeah, and that the blood vessels start gaining size because they start forming the bone marrow, where is all the blood cells originate. And you can see that in addition to that, yeah, yeah, that the formation of the trabeculae, you see the osteoblast still outlining those bones. So in this section, in this third step, you start forming the trabeculae by um, secreting all this extracellular matrix that later will fuse in and form the entire spongy bone. So this is the last step. Ossification center was already done. Then we calcify it, and then we create the trabeculae of the spongy bone that it fused. So here you can see that the spongy bone is there. Yeah, you have all these trabeculae that fuse. You have the blood vessels that are forming the red bone marrow. In addition to that, on the side you have, yeah, no more trabeculae. You have compact bone there, and surrounding that you will have this periosteum. So you remember that I told you the mesenchymal uh, mesenchyme was condensing because he was going to start forming this connective tissue that surround the bones. So remember, the last step is the development of the periosteum, and the mesenchyme in the periphery, at the periphery of the bone, develop into periosteum. Okay, so with this, we finished the first type of embryonic or fetal type of growth. So we now know how the loose connective tissue can turn into a bone. Now we're going to go into the endochondral ossification that shows how the cartilage is also able to form a bone. In the endochondral ossification, we replace the cartilage, yeah, by bone. Okay, and please remember that this happened in the embryonic and fetus stage. And just to show you the process, I'm going to talk about 6 steps that are in your textbook that you can review it in case that you have a doubt. But the main idea is that in the beginning you're going to form cartilage, and then slowly you're going to start replacing that cartilage by bone. Yeah, not only in the diaphysis but also in the epiphysis. So here we have the first 2 steps of the endochondral ossification. And just to start, I wanted to show you the different components. Here, this is the proximal epiphysis, the diaphysis, and this is the distal epiphysis here. So here in the diaphysis, in the middle, you will start developing the cartilage model. And how you do that? You have mesenchymal cells. You remember the precursor cells. So let's put here mesenchymal cells. And these mesenchymal cells will migrate into that region, and they differentiate into osteoprogenitor cells. As there is no capillaries in that area, the osteoprogenitor cells, instead of forming osteoblasts, if you remember, so they are not, yeah, they are going to form chondroblast. And this, chondroblast. And these chondroblasts later, they will start secreting a matrix. Yeah, so they will secrete a matrix, and that matrix, it will form the chondrocytes that later it will become the model, the cartilage model for future bones. Okay, so remember, first thing that happened, we have to develop the cartilage model. And how we do it? We have mesenchymal cells, and those the same mesenchymal cells will migrate into this diaphysis, and they develop into chondroblasts because there is not capillaries that, so the osteoprogenitor cells will become chondroblasts instead of osteoblasts. And this is how you start forming the the cartilage model. In the second stage, we're going to make this model, yeah, this cartilage model to start growing. And how we do that? We start having cell division. So you can see here that the cells start dividing and dividing. And as a result of all this division, there is more chondroblast, and those chondroblasts are going to secrete more material, and they are going to calcify the matrix, and they will start forming chondrocytes. So uh, for the growth of the cartilage model, what we do is we have cell division, and it will allow the chondrocytes and this to have not only higher number, but start calcifying that the cartilage model. So third step, in the first one, we develop the cartilage model. In the second one, we grew this cartilage model. In the third step, what we're going to do is replace this cartilage model. Why? Because we're going to develop a primary ossification center. So we're going to remove the cartilage and we're going to replace it by bone. And how you do that? You do it because in the diaphysis, you have again here blood supply. So remember, if you have a blood supply, high blood supply, and increase in the blood supply, you're going to turn the cartilage into bone. Yeah, so you are switching the pathways, and here this is exactly what happened. Here you see that the primary ossification center start there. So instead of having these blue areas, all those blue areas in the center, they are replaced by bone. Okay, and so what happened there is that due to the increase in vascularity, the chondrocytes enlarge and they become chondroblasts, and then they died. On the other hand, the increase in vascularity stimulate that if you remember, here we have the osteoprogenitor cells, and now these osteoprogenitor cells will become, instead of becoming chondroblast, not anymore, they're going to become osteoblast. And this osteoblast will secrete again the matrix as we have done it before, and they are going to start making bone. In addition to that, the periosteum, here you have periosteum, before you have perichondrium. As now you start forming not cartilage, you're forming bones, you have a different connective tissue surrounding that area. Okay, so far, to summarize, I told you that we, in the first step, we create and grow the cartilage model. Now we're going to replace it by the primary ossification center. So in the diaphysis, you have this nutrient artery, very important. And if you see in that area, the bone tissue has replaced most of the cartilage. Okay, and now in the following step, we're going to continue this replacement. Okay, in the fourth step, so we took this cartilage model, making it grow. Now we decide to replace it by bone. And here what we're going to do is a sculpt, remove some sections that we don't need of the bone to start creating this medullary cavity. And later that will turn into the bone marrow. So before showing you all how this happened, I just show you here again the nutrient artery and vein. So they start going here and they start moving yeah, into those regions to continue the process of removal um, of the cartilage. Here you can start seeing the medullary cavity. Yeah, so you are removing some bones, and how you do it? Because you have these magical cells that we have been talking before that are called osteoclasts. The osteoclasts are going to break down the bone and they are going to form this medullary cavity. Here you still have this area, if you see it very clear there, you have the periosteum, much better differentiated, and on the top you still have the cartilage that later will be replaced by bone. So just to finish this section, I told you that we developed the medullary cavity and how we did that because we have a bone breakdown, and that bone breakdown is done by osteoclasts, and these osteoclasts are going to make this cavity where you're going to storage lipids or you're going to form, for example, different blood cells. Now we're going to talk about the fifth step. In the fifth step, you have the development of a secondary ossification center. So if you remember, we have the first ossification center here in the diaphysis, and it was because we have the artery, the nutrient artery, sorry, the nutrient artery that went there and changed the pathway, and the chondrocyte disappeared, all the cartilage model disappeared and turned into bone. Now that we're going to do a secondary ossification center, we will need a different blood and a different blood vessels, and those are these ones, the epiphyseal, artery, and vein. And if you see it here, they are going there, and in those places where there is blood supply, they will start changing again the cartilage model, and they are turning it into bone. Okay, and this is here. This is, well, this one was the primary or the first ossification center. Here we are going to have in the epiphysis the secondary ossification center. So to summarize, we were talking about the secondary ossification center, and I told you that this doesn't happen anymore in the diaphysis. This will happen in the epiphysis, where you have this important epiphyseal artery and vein that they will increase the blood supply and replace the cartilage model. So here we finish. This is the sixth step. So here we have the formation of the articular cartilage and then the epiphyseal plate. So I remind you, in the first step, we create all this cartilage model, we made it grow, and in the following steps, we were sculpting the different parts of the bone, creating different ossification center, and now we go back and talk about cartilage. And why? Because cartilage is important. So here you will have two types of cartilage that are fundamental for the proper functioning of the bone tissue. So first thing, you have this articular cartilage that is formed of hyaline cartilage. This one are important to reduce the friction between bones. And the second one is this epiphyseal plate. This epiphyseal plate is also important too because, as we are going to talk later, as you grow in length, yeah, and become taller and taller. So when you were a kid, you let's draw a kid. So just imagine that you start getting taller and taller and taller until you become a teenager. Yeah. So one way to predict if you can become even taller or not is go to the doctor, and the doctor will look at your epiphyseal plate. Yeah. So if as you grow taller, you start removing all these, reducing and reducing the epiphyseal plate. Yeah, this area start getting smaller and smaller. And if you are going to keep growing and growing, you will have more space there. Yeah, so if you're going to, up to this point, that this epiphyseal plane is going to become an epiphyseal line. That is the time when you stop growing, okay? And we will talk more about that in the following section, that we're going to talk about how bones go in length and in width. Okay, so
just to finish, I told you that in the sixth step, we go back and talk about the cartilage, yeah? And we talk about the articular cartilage and also the epiphyseal plate. And remember, both structures consist of the hyaline cartilage. So just to finish, here we have again the picture that's showing your textbook of a 12-volt fetuses. And here, as we have been talking, we have regions that are calcified and uncalcified. So we talk in the beginning, if you remember, that we have this intramembranous ossification that happened, if you remember, in the skull, clavicle, and that is much faster. So here you can see it in reddish. So those red areas represent bones that are calcified. In addition to those areas, we have also bones that are calcified in a different way. The one that we just saw here, that they come from cartilage, that it is calcified and form bones. So here you have the ribs, the long bones of the legs, the arms, yeah, and those one also, if you see, they are staining like bones. In addition to that, you have clear areas that are cartilage, that are regions that are uncalcified. So I told you that here in these regions, there is mainly form of cartilage, and they will be replaced, these epiphyseal plates, by bone as you grow, and as when you finish growing, they, instead of epiphyseal plane, they will be called epiphyseal lined. And of course, there will be regions that they will not be calcified, like the nose and the articular cartilage that are in between bones that reduce the friction. Okay, I hope that this was a good summary slide of everything that we saw. If you have any question, just let me know, and I hope that you have a good day.